Assalamu alaikum. Hello everyone, my name is Maria. Today I'm going to talk about glycolysis from chapter 8 in biochemistry. So to begin with, this chapter will discuss the glycolysis, which is the breakage of glucose to get ATP, gluconeogenesis, which is the synthesis of glucose, pentose phosphate pathway, which is the conversion of a hexose to pentose, and the glycogen metabolism. Okay, so our body has three main sources of energy: carbs, fats, and proteins. Carbs are the most accessible one. That's why our body relies on it more. Okay, now before we actually start, we need to clarify these three terms: metabolism, catabolism, and anabolism. Metabolism is a set of reactions that control the homeostasis of the cell. So homeostasis means the normal state maintaining. The amount of ATP, for example, needs to be maintained. Our cell cannot be active or functioning normally if it doesn't have enough ATP, right? Or for example, it can't just have a lot of ATP, excess amount of ATP. So we need to sustain life. That's why this homeostasis need to be controlled by what? By metabolism, which is what? Set of reactions. Okay. Catabolism or oxidation is basically energy production. So we are here mostly breaking molecules to get energy. Anabolism, which is reduction, basically we are synthesizing molecules. We are investing more energy to get a molecule. Okay, now we are going to talk about glycolysis. Glycolysis is anaerobic pathway that splits glucose into two pyruvate units. The captured energy is 2 ATP, 2 NADH. And we need to understand that when ATP is directly synthesized, that means substrate level phosphorylation occurred. Yani we are getting on the same level ATP, okay? When the ATP is not directly synthesized and we need to get it from NADH through electron transport chain, for example, this is called oxidative phosphorylation. Okay, and the aim of glycolysis is to produce energy, and it happens in the cytoplasm. So does it happen in cells that lack mitochondria? Yes, it does. And also glycolysis is considered to be incomplete oxidation. What does that mean? Oxidation is when we are breaking the molecule and getting from each carbon CO2. So for example, if we have a molecule that contains 8 carbons, we need to have 8 CO2. Or 6, we need to have 6 CO2 in the case of glucose. But here, this will not happen. So no CO2 will be released, alright? But we will break the molecule and we will get energy. Okay, so now we will have here our pathway. Okay, so here we have our pathway, which starts with glucose, ends with 2 molecules of pyruvate. So what we need to know about this pathway? We need to know all the reactions, all the enzymes, the products, and the regulatory steps, which are 1, 3, and 10. And we need to understand that when we have the step reversible step, that means this step is not under regulation. Okay, so now glycolysis, 10 reactions divided into 2 stages. The first stage is investment, the second is the payoff. So to begin with, reaction number 1, glucose converted to glucose 6-phosphate. Basically, we add phosphate to carbon number 6. Okay, how it's done? We have ATP, ناخد من ال ATP a phosphate group and we add it to the glucose. So in this way, we will trap the glucose inside the cell, so it will not leave. Okay, يعني basically we lie to the glucose. We say, come, we will give you something. We give him phosphate. Yes, you can't leave. Hello. Okay. So tip number 1, kinases involve the use or production of ATP, and magnesium also will always be required as a cofactor. Let's move on to reaction number 2. Reaction number 2 is simple. Conversion of glucose 6-phosphate to fructose 6-phosphate. So what's the relationship between these two? Isomers, right? Fructose is a ketose, glucose is an aldose. So what enzyme catalyzes this reaction? Phosphoglucose isomerase. Okay, and now tip number 2, understand the difference between isomerase and mutase. Basically, we'll talk about mutase later. بس isomerase is when we have two different structures يعني مثلا ketose and aldose. Mutase is when we are changing the location of a group يعني مثلا we have phosphate group that was on مثلا glucose 6 phosphate will be converted to glucose 1 phosphate, right? We just changed the location, we didn't change the whole structure, right? Okay, I hope that made sense. Now reaction number 3, it is the most regulated step in glycolysis. It is the commitment step. What does that mean? أول ما this step occurs خلاص this molecule will be committed to glycolysis. Okay, so this reaction will be catalyzed via the enzyme phosphofructokinase 1. Phosphofructokinase 1 will add phosphate to carbon number what? Number 1, as the name suggests. So to carbon number 1 we will have a phosphate group attached. So yeah, phosphofructokinase 1 is an allosteric enzyme. يعني it has activators and it has inhibitors. By the way, in glycolysis we have 3 allosteric enzymes: hexokinase, phosphofructokinase 1, and pyruvate kinase. We will talk about that later. But for now, understand that activators of phosphofructokinase 1, AMP is one of them. AMP is an indicator of low energy. فيعني when the cell needs energy, it will activate phosphofructokinase 1 to do glycolysis, which will produce energy, right? And inhibitors are, one of them is ATP. Basically, when the cell has a lot of ATP, why would we need glycolysis? And yeah, then we have reaction number 4, which is simple, the cleavage of fructose 1,6-bisphosphate to acetose and aldose, which are dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. What enzyme? Aldolase. Okay, so aldolase is basically like scissors, just cuts the structure, and yeah, that's it. Okay, now reaction number 5. In reaction number 5, we will have isomerization. This dihydroxyacetone phosphate cannot be used as it. We will need to convert it to glyceraldehyde 3-phosphate. By what enzyme? By triose phosphate isomerase. Triose stands for triose, okay, because we have a carbohydrate or a sugar molecule that is made up of 3 carbons. Phosphate because we have 1 phosphate, and isomerase basically because it is an isomerization reaction. So a ketose was converted to an aldose. And yeah, that's it for investment stage. Now we have reaction number 6. Reaction number 6 is when we will have our first gain, which is 2 molecules of NADH. And in chapter, chapter 6, which is enzymes chapter, we talked about that NADH, where when we see NADH, we should understand that mostly we will have dehydrogenase enzyme. So here we have glyceraldehyde 3-phosphate dehydrogenase, as simple as that. So we have glyceraldehyde 3-phosphate, we will give to this molecule inorganic phosphate. We will lie to this molecule, we will say, come, get phosphate, and you will give us a hydrogen molecule for the NAD, so we will get our NADH. So by the end, we will have how many molecules of NADH? 2 molecules. This is our first gain, again, but it's not substrate level phosphorylation. Okay, then we have reaction number 7, where we will have our first ATP. هون احنا رح نعوض what we lost. Okay, so glycerate 1,3-bisphosphate will be converted to glycerate 3-phosphate. How? الفوسفات الموجودة on carbon number 1 will be transferred to ADP and get us ATP. How many ATP we get in this reaction? 2 ATP molecules. Don't forget to multiply by 2. Alright, then we have reaction number 8. Reaction number 8 is simple. All what will happen is that we will move the phosphate from carbon number 3 to carbon number 2. What enzyme we will use? Mutase. Why mutase? We said mutase is used لما احنا we are changing the location of the attached group. Okay, and this reaction has an intermediate, which is glycerate 2,3-bisphosphate. Basically, this reaction is a 2-step addition elimination cycle. كيف يعني? Basically, we had glycerate 3-phosphate, صح? We add phosphate to carbon number 2 and we remove the phosphate from carbon number 3. So addition and elimination. Then reaction number 9, we will have the enzyme called enolase that will do dehydration reaction on glycerate 2-phosphate and give us water and phosphoenol pyruvate. Okay, so just say beep. Okay, beep يعني like an alert from because of water removal. يعني I'm just trying to simplify stuff. Okay, then we have reaction number 10. Reaction number 10, we have phosphoenol pyruvate that will be converted to pyruvate. How? But simply we will remove this phosphate. So this phosphate will go to ADP and give us ATP. So how many ATP we will gain? 2 ATPs, right? And this reaction catalyzed by the enzyme pyruvate kinase. Kinase يعني what cofactor is needed? Magnesium. Okay, so we have what? 2 ATP produced. So totally how many ATP we produced for what is the net of ATP? 2. I wrote it here. So the regulatory irreversible steps 1, 3, 10. Okay, so yeah, we have here this regulatory step, which is okay. So what happens to pyruvate? Basically it depends where it is. فمثلا if we have aerobic environment, it will proceed to citric acid cycle and electron transport. If the environment is anaerobic, it's either alcoholic fermentation in the yeast and production of ethanol, or a homolactic fermentation in muscles, red blood cells, and certain bacteria. So we will get lactate. Okay, so why fermentation occurs here and here? Basically, it will occur to regenerate NAD that will be needed where in glycolysis. In what step? Step number 6, which was the step where we had the first gain. Okay, so now we are going to talk about regulation of glycolysis. We have allosteric enzymes, which are hexokinases, phosphofructokinase 1, and pyruvate kinase, hormones, and glucose-induced gene expression, and AMP-activated protein kinase. Okay, so to begin with the last one, AMP-activated protein kinase in skeletal muscle and heart, we will have the recruitment of glucose transporters by the plasma membrane. Okay, in the heart we will have activation of phosphofructokinase 2. When we have activation of phosphofructokinase 2, we will have fructose 2,6-bisphosphate, which is an activator of what? Of phosphofructokinase 1. Okay, and when it's done via the glucose-induced gene expression, so mainly when we have a lot of glucose, for example, glycolysis can be initiated via gene expression. Okay, via hormones, basically we have glucagon and insulin. Glucagon is produced by alpha cells in the pancreas, and insulin by beta cells. Glucagon, what's the aim of glucagon? Increase glucose in the blood. So what it will do? It will inhibit glycolysis. Insulin, بالعكس, it will activate glycolysis because it will reduce the levels of glucose in the blood. So basically what it does, insulin will activate phosphofructokinase 2, which is an activator of what? Phosphofructokinase 1, an activator of glycolysis. Glucagon will activate the phosphatase of phosphofructokinase 2. So fructose 2,6-bisphosphate will be reduced, so phosphofructokinase 1 will not be activated. Okay, and via allosteric enzymes also is a type of regulation. Basically we have these enzymes, and this table summarize it up. I won't go deeply on it. So yeah, I think that's it for glycolysis. I hope everything made sense. Feel free to ask questions. And here I have a question for you. So, complete degradation of glucose in anaerobic conditions will produce what? Okay. I will answer the question now. So basically when it says complete degradation, that means that glucose will not be there. We will have what? Pyruvate, right? Okay. This pyruvate is present in anaerobic conditions. So what? It will be transferred to what? Lactate, right? Okay. When we are talking about glucose, that means that this 6-carbon unit gave us lactate, right? So how many lactate? Lactate is 3 carbon. So it will give us how many lactate? 2 lactate. Glucose glycolysis net of ATP was how many ATP? 2 ATP. How many NADH? 2 NADH, right? But later, because of the fermentation in anaerobic environment, this NADH will be transferred to NAD+, okay? So the answer is B. I hope that made sense, and thank you.